Oh, Sushmanth.
Just a minute. I'll set up my laptop.
All right.
Yeah. Okay. Hi, Randy.
Hi, how are
I
you?
just want to I just want to show you the demo of like how the thing works in CellSAM.
Okay.
Okay. In last week meeting, due to I was in outside, I couldn't show you the complete project in a clear way. So, Bradley, what I did is, let's open my report. Bradley, I will start with where I have started this whole project. Coming to SAM segment anything model, it's a promptable segmentation model, and it was the best model till now compared to all different models released now. So it has two types of prompts. One was bounding box, one was the point prompt. So I tried to fine tune it on microscopy image dataset. The dataset belongs from cell tracking challenge.net. I took these datasets. I have trained it all the models, model organisms which are there here. Let me show you. All these 3D plus time datasets are there, right? I have trained on all of them. I haven't left anything. Luckily, the model was performing well. So let me show you with an example how this thing works. Uh, and while fine tuning it, I have required a lot of GPUs actually. Luckily, I bought it through money, and it doesn't cost me much.
Okay.
Cost me around a couple of bucks due to my student ID and stuff. So yeah, here is the model. I'll just show you with an example with a bounding box prompt and the point prompt. And these files are running just a minute. Yeah. I have trained the model on the lowest size model, 375 million parameters. But there are other models also which are bigger than this. They contain of around 1 billion parameters, etc.
Wow.
Uh, so I have trained with on the smallest model. Uh, okay. I need to upload an image. So let's take this one. Uh, okay. This is how we can use this. If you give us a bounding box over this cell, see it has been segmented. Like
Okay.
this only we can give a bounding box to each cell, so it will segment it properly. You may think Bradley like we have a DevoLearn, why do we need this? Because when we are using an electroscopy images of live cell fine tuning, okay, we are taking a data set and training it and feeding it after the data set is acquired. But if we have the lifetime data set, like something right away taking from an electronic microscopy images and feeding it to the model, at that time this model will work very fine because if you want to analyze a protein structure or a cell of a particular cell or an organelle example, it can segment so perfectly, and this thing works badly, way perfectly. It works than any other model. Sorry, you need to give bounding box correctly. It will segment it. Actually, segment anything. See, it has perfectly segmented all the cells, and I have checked with other things also when the cells are densy or when the cells are very low. It is giving better accuracy if we give a correct bounding box around the cell. So this can be used in live cell fine tuning. It is bounding box method, and another was point prompt method. Let me run this thing also.
Okay.
Uh, okay, I will show with these examples. This is the original image, Bradley. So when we give this thing, okay, so it's running. Okay, if we give a point prompt over a cell, this example over here, it has segmented it properly. I have tried more the bounding box prompt, Bradley, due to this GPU issues. See, we have the free GPUs from Collab and Kaggle also, but they are not sufficient because SAM is very big model. To train them, we need a lot of GPUs. So I was able to train bounding box perfectly, but when it came to the point prompt, I couldn't train it as I expected. So the results were not good. It's still giving a better results compared. See, okay. This is what happened with the point map, but it is working very fine with the bounding box. If you can see here, it has segmented properly each and every cell.
Yeah.
And this could be extended for the next GSoC project, like improving this model. What I have done by using by applying the learning rate schedulers, etc. I haven't applied all of them, and we can create a demo in Hugging Face Space or Render also. Otherwise, not next GSoC project badly. I can work from November or start contributing to it from November 15th, and this is what I did. The project looked very simple, but understanding this model and implementing was very hard.
Yeah.
Okay, we are not only the first one to work on this thing. There is another repo also, MicroSAM. These people are also working on this thing using segment anything model for exactly for microscopy images, but they are they are little way ahead than us. They have started their project on the live cell fine tuning, but we have a perfect structure to fine tune this thing. See, um, they are doing on the, I don't know which cells are these, but they are doing like this. Our model also works like this only, Bradley. By giving the bounding box prompts, it will segment it properly.
Yeah. Yeah. So
Okay,
you
and
just use
coming,
either a point or a box to
yeah, exactly,
as the prompt. Yeah.
exactly. These are all live microscopy images, which are fed from an electron microscopy to
Yeah.
this one. So this is what I have done, Bradley. But these people haven't done these things. I have added the multi GPU support because most of the people can't afford. I have done, sorry, one GPU training and multi GPU. If we have the multiple GPUs, like if we have around four GPUs or etc., they can use this model and train better well. So I have added this feature, like the training can be separated in four different GPUs, and I have added the extension point prompt method and inference files. Tutorials are like how to use it, not only for C elegans, but it could be extended to other model organisms also. And this is what I have done, Bradley. Right now, I was working on the paper. I have added the model checkpoints over here. like whatever checkpoints I have, bounding box checkpoint and point from checkpoint, and dataset which I have trained on the CLGAN's dataset, a sample dataset I have added it Bradley.
All right.
So if we, I think we can extend this project further more, and we can take it up to a tool which can segment the cells on the live, analyze the protein structures, etc. This is my aim, and I was working parallelly on DevoNet also, Bradley. I hope I haven't completed it due to this GPU issues, and only I couldn't try because whatever the data set I have right now, it is a 3 GB data set, and I need around 4 GPUs to train it. Those are V100 GPUs I need. But in Collab subscription, I can get only one GPU. I can't ask for like multiple GPUs. The
Right.
subscription doesn't provide me those many GPUs. So I have approached my college for the GPUs. They liked my project, so they told like, yeah, you can take the GPUs. But due to some PhD students are already using those GPUs, I couldn't get in. But next couple of weeks, I will get access to that also, and I will push code. I will train the DevoNet model, and I will host it in the DevoLearn GitHub. Right now, I have kept the library as a private because the work is going on, right? I don't want someone steal it or something like that. So I have kept it private. And after the complete code and the model is trained, I want to make it as open source.
All right. Yeah. Yeah.
Is that okay? And the model works very fine, Bradley. I hope you give a look on this these people's work also.
Okay.
Uh, my code and these people code are completely different. I have done what I could do, everything. I want to contribute more and go analyze the protein structures. Not only we are, I think right now DevoLearn is meant for CL GANs only. If we could extend it for the protein structures, bacteria analysis, etc., could be more useful. I think so. I don't know about the underhood of the biology, but this could be extended.
Yeah. Yeah. Definitely could. All right. Yeah, that looks great, and I think we can find a lot of uses for it.
Yeah.
Yeah.
And those are my updates, Bradley. Last week I couldn't show you the demo, but in coming weeks I want to work on these things. I want to host it in the Hugging Face spaces or create a web tool so any biologist can use it, or either I will work on the DevoNet model and train it if I get the GPUs. I have I have found out the way to host it on the web as a tool. We can decrease the model size and then and compress it, and we can use it as a tool. Host it in the Hugging Face spaces or DevoLearn web is there, right? There we can host this thing. But
Yeah.
I need little time to work on this thing to learn the stuff, to compress the model, etc. Thanks for that. And these are my updates, Bradley.
Okay.
And I will work the next couple of weeks, and I will attend the meetings also,
Yeah.
and I will let you know if I can make further changes. And could you give my evaluation in the GSoC? I think there is a deadline on October 31st
Yeah.
from your
I'll
side.
take care of it. I'm gonna probably do it tomorrow. So.
Okay, thanks. I hope you are happy. I just want to see how this thing goes. I'm trying to learn and implement as much as I can.
Oh yeah,
And
this is great. This is great work. Yeah. Thank
okay,
you so
well, I
much for
will.
your efforts. Yeah. And we'll keep working on it. And yeah, it looks like it's a great. You've got not only one edition but several additions to DevoLearn. So this is great.
Thanks, Randy. Thanks. So hope I
Yeah.
can do much more better. I will start contributing more from next week. I have exams right now.
Yeah.
So tomorrow onwards I have exam. By next Monday I will start contributing again.